Hello, this lecture will go over Chapter 39, Assessment of the Hematologic System. Concepts that will be covered include clotting and perfusion. Your slide here gives you definition of perfusion and clotting. Perfusion being total arterial blood flow through the tissues, peripheral perfusion, and blood that is pumped by the heart, central perfusion. Clotting is a complex multi-step process by which blood forms a protein-based structure or clot. In terms for the anatomy and physiology review, these should all be very familiar to you from your A&P classes. We'll hit on a couple of things here. So bone marrow is where blood stem cells are produced, right? And then the stem cells determine their path and what type of cells they'll end up developing into. So bone marrow first produces blood stem cells. Those are immature, unspecialized, undifferentiated cells that are capable of becoming any type of blood cells. Then the next stage in blood cell production is committed stem cell. Committed stem cell enters one growth pathway and can at that point specialize into only one cell type. For example, erythropoietin is a growth factor specific for red blood cells. You have a little figure on page 796, figure 39-2, that shows the different pathways from a stem cell to the different types of cells that it can be matured into. So just review that basic A&P review. Under blood components, those plasma proteins, albumin is important to maintain osmotic pressure of the blood. Globulins have many functions, such as transporting other substances as antibodies, protecting the body against infection. Fibrinogen is activated to form fibrin, which is critical for clotting. Red blood cells, you guys should be thoroughly familiar with red blood cells and white blood cells and platelets. I'm going to let you review the text on your own to go over those cells, because I think you probably feel pretty comfortable with them at this point. If you don't, read them, go over them until you do feel comfortable with them. Under accessory organs of blood formation, the spleen. The spleen has three different types of tissue: white pulp, red pulp, and marginal pulp, that help balance blood cell production with blood cell destruction and assist with immunity. White pulp is filled with white blood cells and is a major site of antibody production. As whole blood filters through the white pulp, bacteria and old red blood cells are removed. Red pulp is the storage site for red blood cells and platelets, and marginal pulp contains the ends of many blood vessels. The spleen destroys old or imperfect red blood cells, breaks down the hemoglobin and release from these destroyed cells, stores platelets, and filters antigens. Anyone who has had a splenectomy, has had their spleen removed, has reduced immune function and increased risk for infection and sepsis. The liver is important for clotting. The liver produces prothrombin and other clotting factors. Proper liver function is important in forming vitamin K in the intestinal tract, which then goes on to play a role in other clotting factors. Large amounts of whole blood and blood cells can be stored in the liver, and the liver also stores extra iron with the protein ferritin. Hemostasis and blood clotting. So this starts on page 798. I know in A&P you reviewed blood clotting. I want you to read through this section on your own and review it again. We got a little video here as a quick review. Let's see if I can get her going. Okay, let's get back to our PowerPoint. All right, so again, the process starts with platelet aggregation. On page 798, you have an italicized line. These platelet plugs, bear with me a moment. All right, sorry about that. So again, platelet aggregation is step one. The platelet plugs are not clots and only last a few hours, thus cannot provide complete homeostasis, but can only start the hemostatic process. When too few platelets are present, clotting is impaired, increasing the risk for excessive bleeding. Blood clotting is a cascade triggered by the formation of the platelet plug, which then rapidly amplifies the cascade. Your text talks about intrinsic factors and extrinsic factors that you can review. During the cascade from the formation of a platelet plug to the formation of a fibrin clot depends on the presence of specific clotting factors, calcium, and more platelets at every step. Clotting factors are inactive enzymes that become active in a sequence. The last two steps in the cascade are the activation of thrombin from prothrombin and the conversion by thrombin of fibrinogen into fibrin. Only fibrin molecules can begin the formation of a true clot. Fibrin clot formation is the last phase of blood clotting. Fibrinogen is an inactive protein made in the liver. The activated enzyme thrombin removes the end portions of fibrinogen, converting it into active fibrin that can link together to form fibrin threads. The fibrin threads make a mesh-like base to form a blood clot. After the fibrin mesh is formed, clotting factors tighten up the mesh and make it more dense and stable. More platelets stick to the threads of the mesh and attach other blood cells and proteins to form an actual blood clot. As the clot tightens, the serum is squeezed out and the clot formation is complete. You can review page 799, figure 39.4 is a nice diagram over blood clotting. It also indicates there where different types of medications interfere with blood clotting, whether it be platelet inhibitors, warfarin, fibrinolytics. So while you're reviewing that figure, go ahead and note when those medications are affected, how they affect that process. Anti-clotting forces. Whenever the clotting cascade is started, anti-clotting forces are started to limit clot formation only to damaged areas, so normal perfusion is maintained everywhere else. So this is basically the body's way of making sure the clots only occur where they're needed. Fibrinolysis is a dissolving of the fibrin clot edges with special enzymes. The process starts by activating plasminogen to plasmin. Plasmin then digests fibrinogen, and prothrombin, controlling the size of the fibrin clot. These actions prevent clots from becoming too large or forming in areas where clotting is not needed. The hematological changes associated with aging, I'm going to let you review on your own. There's a little chapter and then a nice diagram. A big thing to know is that as bone marrow ages, it produces fewer blood cells. Platelet count, though, does not change. So red blood cells, white blood cells go down. Platelet count does not change. Lymphocytes become less reactive to antigens and lose immune function. Antibodies levels and responses are lower and slower in older adults. The white blood cell count does not rise as high in response to infection in older adults as it does in younger adults. These assessment part of the chapter, I'm going to let you look over on your own. A lot of the medications talked about under patient history, we've already talked about in lecture and you're familiar with. When you're studying, focus on those boxes and anything italicized. Make sure you're not overlooking those. So we're going to fast forward to these diagnostic assessments. A peripheral blood smear provides information about the size, shape, proportions of different blood cells within the peripheral blood. A CBC is not new to you. It includes red blood cell, white blood cell counts, hematocrit, hemoglobin. Let's see here. I'm going to go on to the reticulocyte count. Is helpful in determining bone marrow function. The reticulocyte is an immature red blood cell that still has its nucleus. An elevated reticulocyte count indicates that red blood cells are being produced and released by bone marrow before they are mature. An elevated reticulocyte count is desirable in an anemic patient or after hemorrhage because this indicates that the bone marrow is responding to a decrease in the total red blood cell level. An elevated reticulocyte count without a precipitating cause usually indicates health problems such as polycythemia vera, which we'll talk about later in the class. Platelet counts you guys are familiar with. Hemoglobin, electrophoresis, we'll talk about when we talk about sickle cell disease. It detects abnormal forms of hemoglobin. Leukocyte alkaline phosphate is an enzyme produced by normal mature neutrophils. If it's elevated, it could indicate infection or stress. Combs test is used for blood typing. The direct test detects antibodies against red blood cells. Indirect Combs test detects the presence of circulating antiglobulins. And the serum ferritin test measures the amount of free iron present in plasma. Transferrin is a protein that transports dietary iron from the intestines to cell storages. Total iron binding capacity, or TIBC, increases when a patient is deficient in serum iron and stored iron levels. You got a chart on page 804. This is important to note. There are specific levels here that I want you to know for testing purposes. You are required to know the reference ranges for red blood cell count, hemoglobin, hematocrit, white blood cell count, platelet count. INR we've already talked about. You know that a therapeutic INR level for a patient on warfarin is 2 to 3. So we'll leave it at that for now. I'll let you look up those other, again, a lot of these we've talked about, these tests that measure bleeding times. You can look at those on your own. X-rays. The bone marrow aspiration and biopsy. It's on page 806. A bone marrow aspiration and biopsy can help evaluate a patient's hematologic status when other blood tests show abnormal findings that indicate a possible problem in blood cell production or maturation. Including the production. Results provide information about all bone marrow function, including the production of all blood cells and platelets. In bone marrow aspiration, cells and fluid are suctioned from the bone marrow. In bone marrow biopsy, solid tissue and cells are obtained by coring out an area of the bone marrow with a large bore needle. This requires the provider's prescription and assigned informed consent.